Okay, so the title of my talk that I started with, several weeks ago when we were putting the program together, is Material Properties in Biomechanics. And actually, I don't know if it was a good thing or a bad thing. Oh, let me just minimize this so you can see the slides. Um, but I decided to Google material properties in biomechanics, and this is what I found. Mechanical characteristics of bone, cartilage, and tendons. And I was like, what? No muscles? So I had to adjust my title a little bit. So what I'm actually going to be talking with you about today is material properties in muscle mechanics. So let me just say at the outset, I guess I can see the chats do pop up for me. So if you have questions along the way, please do, either put them in the chat or, this should be very informal and as much discussion as possible as we go along. So please feel free to just turn on your microphone and interrupt me and ask a question. So I want to particularly, thank all the people who've contributed to the things that we've been doing in my lab, as well as the funding agencies. But I particularly want to call out Rick Kassler from MIT, who has really been a major collaborator in the work that I'm going to talk about today. So, how I got started in being interested in biomechanics, and some of you who've known me for 20 plus years know this, was studying how frogs and toads, like the one shown here, catch bugs. And if you've ever tried to catch a fly, I guess maybe it was the Karate Kid could do it, but most of us can't catch a fly with our bare hands. And when amphibians have these projectile tongues that they can project from their mouths incredibly fast, and it turns out that their jaw opening muscles are relatively short. They can never be actively stretched because that they're at their longest length. Um, uh, when the mouth is fully closed, and there's a kind of a catch mechanism so that the jaws open extremely rapidly, but very short tendons. So these muscles themselves must, store enormous amounts of energy. But where in a muscle energy is stored and how this might happen remains unclear. So, but as I'm going to tell you in the end of my story today, it'll all come back to material properties of muscle. So this is where it started and where we'll start today. And this is an outline of, what I want to talk with you about today. So viscoelasticity is a foundation of power amplification, and that can be whether you're a muscle or a tendon, or even sometimes animals with flexible bones can actually get some power amplification from their bones. The basis of viscoelasticity is springs and dampers. So we're going to really start simple right from the beginning. And we'll talk about springs and dampers and types of simple viscoelastic models and their properties. We'll talk about different experiments that can be used to measure viscoelastic properties and some early work that was done in the mid-1990s. And then I'll talk a little bit about my current work on muscles as tunable viscoelastic materials, and I'll show you some examples from some studies we've done on mouse and frog muscles. And then this last hot off the press part is going to be moving from viscoelasticity and viscoelastic models to understand muscle power and energy storage. So we'll talk about power amplification and dissipation, and we'll talk about energy storage and recovery. So for those of you trainees who are completely new to this field, there will be math, very simple math, just, like plain old algebra. I don't think we even have any calculus in the talk. I think I avoided derivatives and integrals. And there will also be some physics. So please do interrupt along the way if you have a question or something isn't clear, because if you're not getting it, there's probably at least 5 other people on the Zoom call who aren't getting it as well. So let's start with simple springs. So we have a linear spring, we can hang weights of, varying masses of varying weight from the spring. And we see that the force exerted on the spring by the mass displaces the spring by an amount X. And it turns out that for linear springs, we have Hooke's law, where the force is equal to the spring constant K times the displacement X, right? And the spring constant is a characteristic of materials. So stiffer materials have a higher spring constant, and more compliant materials have a lower spring constant. We can also write this in a non-dimensional form where sigma is a stress, the E is the elastic modulus, and I should say, and epsilon is the strain or the change in length. And for many materials, they will actually have sort of a Hookean behavior at small strains, but eventually, I don't know if you played with slinkies when you were a kid, but I used to play with slinkies and I always broke them and they always got stretched beyond their limit where they yield and they no longer return to their original shape. So at some high strain, we can, break the spring and it won't have a Hookean behavior anymore. And the nice thing about, that as the linear springs, or any springs really, as I'll show you in a minute, is we can scale these relationships from the whole muscle level down to, an individual thick filament that might be interacting with 6 tighten molecules and 3 thin filaments in a muscle sarcomere. So all of these viscoelastic properties are going to scale nicely over large ranges of stress and strain. And so the next kind of, simple viscoelastic property is, the viscous forces. And in the simplest case, we have what's called a linear damper. So damping is usually much less familiar to my students in my biomechanics class than is the spring constant. And so when we think about a damper, we could think of a cylinder filled with fluid and a piston. And so if we apply a force or a stress to the piston, we'll compress the fluid. The piston will move downwards in this case. The fluid will be displaced. And what happens is that the force that we require to displace the fluid is going to increase not with the strain, but with the strain rate. So we have a damping force that's equal to the damping constant times the rate of displacement, which here is denoted by x dot. And we can also have non-dimensional equations for the damping. So we have a non-dimensional damping modulus, just like the elastic modulus. And we have the stress then is equal to, the damping modulus times the strain rate. And one example I like to think about, and I don't know if this one, I gave you the example of the slinky before, but an example of viscous forces is like when you're trying to remove, snails from the basil in your garden, and the faster you pull, the more force the the snail exerts, it's hard. The faster you pull, the harder it is to pull the snail off your basal. But if you pull really, really slowly, you can easily remove it, and the snail can crawl very slowly over your basal leaves while eating them. So now what we can do is we can take these simple elements, springs and dashpots, and we can build up many different models of different viscoelastic materials, each one with unique characteristics. I mean, we can make these more or less complicated. So the simplest two-element viscoelastic models are called a Maxwell model after the famous physicist Maxwell, which is a spring and a damper in series. Or we can have a Kelvin-Voigt, also famous physicists, a Kelvin-Voigt model of a spring and a damper in parallel. We can build up 3-element models, 4-element models, 5-element models, n-element models. We could have, n springs and k dampers in any configuration. And in principle, we could analyze we could write a math model that would predict how these different viscoelastic materials will behave when we deform them. And I just want to point out a couple of the more common ones. This is kind of like a standard viscoelastic solid here, where you have a Maxwell element in parallel with a spring. A Hill model, it has the same components, but they're arranged in a slightly different way, with the spring outside of the Kelvin-Voigt, or a spring in series with the Kelvin-Voigt element. We can have 2 Maxwell elements in parallel. We can have 2 Kelvin-Voigt elements in series. And we can build up models now where we have, 5 or more elements. And for the purposes of this talk, this is as complicated as we're going to get. Okay. So now we can look at some of the behaviors of the different simple models. So the Maxwell model has a spring and a damper in series. And interestingly, the behavior of this model is that stress in this, so remember that's force divided by area, in the spring and the damper are equal, but the strain is unequally distributed between the 2 elements. And this is our simple constitutive equation. So if we have stress as a function of time is equal to the elastic modulus times the strain at time t. And that would be the stress in the damper, the stress in the spring. And then we have the stress in the damper, which is the damping modulus times the strain rate at time t. The Kelvin-Voigt model shows exactly the opposite behavior. So the strain in the spring and the damper are equal, but the stress is distributed. So the total stress as a function of time is equal to the elastic modulus times the strain at time t plus the damping modulus times the strain rate at time t. So we see already, even in these very simple models, that they behave very differently and distribute stress and strain differently when we deform them. And this next slide shows some simple step response properties of these 2 viscoelastic models. So here we're doing a step in the strain, and here we're doing a step in the force. And you can see the Maxwell model, when we do a step change in strain, we see an initial big response that decays to 0 over time. In contrast, when we do a step response in a Kelvin-Voigt model, we see a really big increase in the strain or force during the stretching, which relaxes very quickly. And what remains is the elastic force in the spring. Likewise, if we do a step change in force, we see a creep behavior in the Maxwell model, and we see this time-dependent increase and then decay in the Kelvin-Voigt model. So, if you just remember this here, we're going to look at how muscles respond to step changes in strain in a little while, and you'll see that there are some similarities and differences between these models and how muscles respond to similar step changes in their length. So what we want to be able to do, or what I want to be able to do, is I want to know, what viscoelastic model is going to best fit those toad muscles during that really rapid mouth opening where they must be storing and recovering energy. And these are the models that I'm going to consider. But in order to figure out which is the best model, I'm going to have to do some experiments. So I don't know if there's any questions about simple springs and dampers and simple viscoelastic models before we go on. But if not, we'll start talking about some experiments to measure viscoelastic properties. So I'm going to talk about 3 different types of experiments. One is a ramp, stretch, and hold experiment. So for example, we might take a muscle and start at a particular length relative to muscle optimal length. We will stretch the muscle with some amplitude and some rate, and then hold that stretch at the final length. Another common experiment that's used to measure viscoelastic properties is sinusoidal perturbations. So if we have a sinusoidal strain perturbation, so we're changing the strain in a sinusoidal pattern over time, and in these sinusoidal oscillations, I'm getting ahead of myself. So we have sinusoidal oscillations, and then the third one is white noise. So we can actually randomly change the length of a muscle. We can have a small amplitude. Here it's 0.4 millimeters, or a large amplitude. Here it's 1.6 millimeters. And we can also band the frequency. So we can say, okay, we're only going to allow the frequency to vary between 0 and 15 hertz, or here it's going to vary between 0 and 90 hertz. So these are the 3 types of experiments that are commonly used to measure viscoelastic properties. So now we can look at some of the advantages and disadvantages of each of these methods. So here we have a ramp, stretch, and hold of a mouse soleus muscle. Time is on the x-axis, and force or stress is on the y-axis. And the blue line is an isometric contraction where we just stimulate the muscle and hold it at a constant length. And then we deactivate the muscle and it relaxes to some resting length. The black case is where we actually completely passively stretch the muscle. So no activation, and you can see what that response looks like. And this response looks actually really a lot like that single Kelvin-Voigt body, if you remember in the previous slide. So we have extra force during the stretch that decays, but it doesn't decay ever to 0. It decays, to what looks like some, force in a spring. And then when we stretch the active muscle, we see even more complicated behavior. So we get a lot more extra force during stretching, actually almost as much again as the isometric force, right, at that before the stretch. That decays with some time constant, which we'll talk about later, and then we deactivate the muscle. And one other thing that I just wanted to point out really quickly, so here we see the ramp stretch, and now we We calculate the stretch velocity, and so the stretch velocity is shown here in orange. So even though we are just stretching at a single velocity, we're probing, changes in velocity and acceleration when we start and stop stretching. So we're really looking at the behavior of the material, in this case, a muscle, over a range of velocities, not just the nominal stretch velocity. So advantages are that ramp stretches can be, or I should say in vivo perturbations can sometimes look quite a bit like a ramp stretch. So they have some biological, physiological relevance. It's relatively easy to perform these experiments at different activation levels and different start lengths. So we can do a lot of experiments with minimal fatigue. And we do have these velocity transients, so we're not probing a single velocity. One disadvantage that's cited in the literature is that the response, or the RAM stretches, produce a pretty complicated muscle response that turns out to be hard to model. Sinusoidal oscillations, as I was starting to tell you earlier, the elastic forces are in phase with the oscillatory perturbation, but the viscous forces are out of phase. And so when we have a viscoelastic material, depending on its exact, damping and elastic moduli, we can get quite a different behavior with some in phase and some out of phase components. So advantages of this method are that it's very commonly used in material science. And so if we wanted to compare muscles to other materials in the exact same experiment, this might be the experiment of choice. Disadvantages are that each activation level and start length has to be probed separately, which requires significant experimental time and can induce quite a bit of muscle fatigue. And periodic oscillations are not as similar in general to in vivo perturbations as ramp stretches are. And then finally, the last type of experiment is this white noise experiment, where we're randomly varying the muscle length over time, and we can band the frequency range at which the changes in length are occurring. So each experiment is done with constant activation and a single start length. We have this continuously varying strain, amplitude, and frequency. And one advantage is that, we can combine multiple frequencies simultaneously, so anywhere within the band that we choose. And that can save having to do a lot of different experiments, but we still have to vary the activation and start level separately. So we have to do a lot of experiments in order to characterize, the length and activation dependence of the viscoelastic properties in a muscle. So I just want to tell you, before we move on, about some very early work. This is a paper by Kirsch et al. In 1994 that appeared in, IEEE Transactions in Biomedical Engineering. And here they used cat soleus. It was an in situ preparation in a decerebrate cat. And so they had three models. The first model is a simple Kelvin-Voigt element like we saw previously. The second model is a Hill model. And that third model is that standard viscoelastic solid. So this model has two parameters. These have three, but they're arranged in slightly different configurations. And they probed these three models, or they tested these three models and their ability to predict the force response of the muscles in ramp stretch and white noise experiments. So here are the results of the ramp stretch experiments. So they have the in situ cat soleus muscle, and they're doing ramp stretches at 3 amplitudes, 0.4 millimeters, 0.8 millimeters, and 1.6 millimeters. And these are the actual force versus time responses of the muscles. And down here, you actually see the fit of the models to the data. And so for the 1.6 millimeter stretch, all of the models fit the data fairly well. But for the lower amplitude stretches, only the more complicated 3-parameter models fit the data. And what you could see is that model 1, which is this thin black line, it relaxes too fast. So it's not holding on to the force long enough to make a good force prediction. Conversely, in the white noise experiments, only model 1 fit the data, and the other models fit the data so poorly that they didn't even show the results in the paper, which is kind of funny. So no models fit both the ramp stretch and the white noise data sets. And so we can see that the single Kelvin Voigt element fit the data pretty well. The force prediction looks pretty good, but when we look at the percent variation in force explained by the model, it was in the range of 80 to 85%, which in the modern era is not very good. We strive to do better. Okay, so now we can, sort of start to interrogate this question: what kind of viscoelastic material is muscle? And based on our ongoing discussion, we can rule some of these models out. So obviously, muscle isn't a simple spring or a dash pot. A single Maxwell element and 2 Maxwell elements in parallel both have the property that the force decays to 0 over time, and we know muscles don't behave that way. And then finally, a good model of muscle should predict force regardless of the experiment we pick. And so we can infer that neither of these 3 models are the best model because they didn't predict all of the data collected by Kirsch et al. In both of the experiments they did. So we're going to assume that like the simplest possible model that's still standing is going to be this 2 Kelvin Voigt elements in series. So now I'm going to tell you about some of the experimental work that we've done recently and the results from that. And so what we did, kind of by accident, in other words, when we collected these data over a series of starting in probably 2010, we didn't intend to use the data in exactly this way, but fortuitously, they turned out to be very handy. So in mouse soleus muscles ex vivo, on a servomotor force lever, we conducted ramp stretch experiments. We had a stretch amplitude of 5% L naught and a velocity of 1 L naught per second. And we did these ramp stretch experiments all across the physiological range of muscle lengths, starting at 0.8 L naught and actually going up to 1.25 L naught in increments of 5 to 10%. We've also done this in frog muscles, I'll tell you in a minute, and the mouse muscles were done at 5% increments and the frog muscles at 10% increments. So at each starting length, we stretch muscles both passively and actively on the plateau of the isometric force. And we have a sample size of 7 to 9 muscles. So these are from 3 Titan genotypes of mouse soleus muscles, wild type MDM, which is a naturally occurring mutant, and this transgenic mouse Titan delta 112 158, and I'll tell you more about those in a minute. And we also did this in the bullfrog, and when I say we, I mean Natalie Holt and Caitlin Manross and Apollo Ibanez. Last summer, we did this as part of the previous IMSI Summer Institute. So let me tell you a little bit about the mouse muscles. So we have this transgenic mouse that was made in Henk Granzier's lab at the University of Arizona. And so the PVK titin is what's responsible for passive muscle tension. And so in this case, the Granzier lab engineered this big deletion in PVK titin, which goes from exon 112 to exon 158, hence the name. So it's missing, more than 1,500 amino acids in PVK, which is approximately 75% of the total contour length of the passive PVK spring in passive muscles. And we also looked at this MDM spontaneous mutant. It has a rearrangement that results in a primary deletion of 50 amino acids, which is very, very small compared to the transgenic mouse. And it's right at the, border between the N2A region of titin and the PVK region of titin, which I'll show you in a minute. But post-transcriptional processing reduces the contour length and therefore increases the stiffness of the PVK1 and PVK2, but you can see in the PVK3, the wild type and the MDMs are very similar. And we know from single molecule atomic force microscopy experiments that PVK1 and PVK2 are passively stiffer than PVK3, and this will become more interesting later on. Here are the frog muscles. As I said, this work was done last summer. Well, this slide shows previous work done by Natalie and Henk Granzier. So Natalie measured the active and passive force length characteristics of these three different frog muscles, sartorius, iliofibularis, and semitendinosus. And you can see the sartorius was the stiffest and developed passive tension at the shortest sarcomere lengths, and semitendinosus was the most compliant. Natalie sent muscle samples to Henk's lab, and he ran titin gels on them. And they found 3 titin isoforms by the migration distance in the gels, the shortest, stiffest isoform in the dark blue, the intermediate length isoform in the green, and the longest, most compliant isoform in the light blue. And so you can see the most compliant muscle has none of the stiff titin isoform, and the stiffest muscle has the most. So we used system identification to try and identify what viscoelastic model best fit all the data from the mice and the muscles from the mice and the frogs. And first of all, I'm going to just show you, but we're not really going to talk too much about the constitutive model for the 2 Kelvin-Voigt bodies in series. So you might remember that the model for the simple Maxwell or Kelvin-Voigt model, Kelvin-Voigt element, those were pretty simple equations, but we're getting, as we build up complexity in the viscoelastic structure, we add a lot of additional terms to our constitutive equation for the strain versus time. So, and we're starting to get into the world of time constants. We'll talk a little bit more about those later. So each Kelvin-Voigt element has a time constant. So there's a tau1 for the first one and a tau2 for the second one. And the taus are equal to, in non-dimensional terms, the damping modulus divided by the elastic modulus. And as some of you will be familiar with and others not, we can use the difference equations to estimate the viscoelastic parameters, but I'm not going to talk too much about that right now. But interestingly, what we found was that for all of the muscles, this 2 Kelvin Voigt bodies in series model fit the data with a really high R squared and really small residuals on the order of 10 to the minus 4th power. And so what we're able to do is from the system identification, we're able to estimate all the viscoelastic parameters, K1, B1, K2, B2, and the time constants tau1 and tau2. And now we can analyze how they change with genotype or muscle type, with activation, with starting length, and using analysis of variance, we can also look at interactions between these main effects. And so here's what the data look like. And so here we're looking at the first Kelvin Voigt element, and this is the one that's closest to the Z disk of muscle, with the K2B2 I'll show being closest to the M line, we think. And so these are the data from the frog muscles. So here we have the starting length from which we stretch the muscle. And here we have the value of the stiffness. And this is in newtons per meter. And notice the log scale. So this is like 4 orders of magnitude here. And you can see that the passive tension varies over, close to 3 orders of magnitude from the shortest length to the longest length. So this is an indication that this K1B1 is an exponential spring. And furthermore, we know that the sartorius has the stiffer tighten, it has a higher passive tension, and it also has the higher values of the elastic, of the spring constant, sorry, across lengths compared to the semitendinosus. And we can look here and see the same data for the mouse muscles, the MDM muscle, and the wild type. And today I'm not going to talk too much about the delta 112, 158, except I could maybe say that this 2 Kelvin Voigt bodies in series didn't fit those. One thing that's interesting is that you can see that the frog muscles produce 20 times more force. They're about 3 times as long, and their cross-sectional areas vary by, I think, like at least a factor of 20. But you can see that the values of their spring constants all fall in approximately the same range. And one thing that's interesting is that we can also scale these down. So we can multiply by the number of sarcomeres in series and divide by the number of thick filaments per millimeter squared of cross-sectional area. And we see that all of these mouse and frog muscles have similar properties even when scaled down to the half sarcomere level. And so we think that the K1B1 unit actually corresponds to this PVK1 and PVK2, and that's because the passive K1 follows the differences between the wild type and the MDM in terms of the MDM and also in the frogs. So the K1B1, is stiffer in the MDM muscles than in the wild type. And also, just the fact that the stiffness is varying over 3 orders of magnitude suggests an entropic exponential spring like Titan. So here to save time, I'm not going to show you the damping constants. I'm just going to show you the time constants. But the interesting thing about this K1B1 unit is the time constants are around 100 milliseconds in the passive muscle, but they increase to up to, a half to a full second. So, and what that means is that these muscles, the active muscles can hold on to force. The force decays a lot slower than in the passive muscles. And the same is for the mice. So we see really, really similar relationships and really, really similar values of the spring and damping constants and the time constants between these frog and mouse muscles. The K2 unit that's closer to the M line is really interesting. And it shows a different behavior from the K1. So K2 is initially less stiff and less damped than the K1, B1. But when we activate, it actually gets stiffer. And in fact, it goes from being an exponential spring passively to a linear spring with a more or less constant value of the spring constant in the active state. And similarly in the mouse muscles. And look at what happens to the time constants and how different they are for the K2B2 than they are for the K1B1. So passively, they're around 10 milliseconds. And when we activate, they actually get shorter. So this K2B2 unit really can't hang on to force for more than a few milliseconds. So this is about 5 milliseconds here. And similarly in the mice. Okay, so just really quickly, here's a little cartoon of Titan. This is the entropic PVK region. We think that the K1, B1 is, I have this backwards. We think that the K1, B1 is closest to the Z disk and the K2, B2 is closest to the M line. And the behavior is between the wild type and the mutant is consistent with these units corresponding to PVK1 and PVK2. And this K1B1 unit is completely absent in the Titan 112, 158 transgenic mouse, and the 2 Kelvin Voigt bodies in series model doesn't fit those data. So we think K2B2 corresponds to Titan PVK3. These results are also consistent with the single molecule atomic force microscopy experiments. And this K2B2 is the only unit that's present in the delta 112, 158 transgenic mouse muscles. So before I spend the rest of my time talking about power and energy, I just want to briefly recap the insights that we think we've gained from the system identification. So we think that muscles are tunable viscoelastic actuators. Passive and active muscles have a similar viscoelastic structure that seems to be based on PVK Titan. Passive stiffness and damping vary with muscle length over 2 to 3 orders of magnitude, which is a signature of an entropic exponential spring. Muscle passive tension and Titan gene expression predict the passive viscoelastic properties. And so muscles, we think, are tunable, variable transmission ratio, viscoelastic actuators that can harvest energy from interactions with the environment, and they can release that stored energy to increase the efficiency of movement. And Rick and I are in the process of developing open source Lagrangian dynamics models based on our experiments that we hope will replace Hill models in simulations of muscle force. I'm just gonna, and so the Lagrangian dynamics models are going to have these properties. They're going to have cross-bridge activation dynamics, including the length-dependent overlap of the actin and myosin filaments, very similar to what's in Hill models now. They're going to have N2A actin binding dynamics. So the N2A part of Titan binds to actin in active muscle and sets the muscle equilibrium length at activation. They're going to actually have thin filament Titan winding. So the cross bridges not only translate, but also rotate the thin filaments, and this produces the variable transmission ratio dynamics. And they're going to have the length and activation dependent tunable exponential viscoelastic response by dynamics that I just showed you is very similar in the frog and the mouse muscles. And from this, as I'm going to tell you in a minute, we'll be able to completely describe the power amplification and dissipation of the muscles, as well as the energy storage, energy dissipation, and efficiency. So this briefly is the model, and I'm not going to walk you through this picture of it, except maybe to say, here's the M line, here's the thick filaments, here the thin filament is kind of in the background here, and here's the Z disc. So a way to think about our models conceptually compared to the Hill model, and also oh no.
Yeah, Kiisa, you froze for about 30 seconds, so if you could restate what you were just saying, that would be great.
Okay. Okay. I got ahead of myself here. Yes. So the bottom line is that everything in a Hill model regarding the activation dynamics we're keeping. But what we're saying is that the cross-bridge forces are not the muscle force, because the cross-bridge forces are transmitted to the Z disc by titin, which actually also changes its structure upon muscle activation. So our model has all of the conventional cross-bridge dynamics, but we're adding on this tunable viscoelastic transmission of forces. Did everybody get that? Okay. All right. So this is the last part and the new part. And I'll try, I hope I'm going quickly enough, but I think we'll have plenty of time for questions at the end. So interestingly, as you'll see in a minute, we can directly estimate the power added by the titin transmission to the system, as well as the energy storage and dissipation. So let's go ahead and take a look at each of those, and we'll start just by doing some simple math to define some quantities. So force is equal to the mass times the acceleration. Work is equal to force divided by displacement. Power is equal to work divided by time, which actually, if you do the math and substitutions, you'll find it's also equal to the force times the velocity. So the positive power amplifies in the transmission, amplifies the cross-bridge forces, and negative power is going to dissipate the cross-bridge forces. And the elastic energy stored by a viscoelastic material is commonly written as 1/2 kx squared, where k is the damping constant and x is the displacement. But by substitution, we can also show that the energy stored is equal to 1/2 the force squared divided by the stiffness. So now that we've estimated our stiffnesses from the system identification, we can directly measure the energy stored in the Kelvin Voigt elements during those passive and active stretches. And it turns out, furthermore, that about 2/3 of the elastic energy stored dissipates in one time constant. So now we can look at some data, and I'm just showing here, we're just getting started on this analysis Rick and me, and eventually we'll do it for every single one of our like 300 and maybe 400 experiments, given all the mice and frogs. But just to give you a feeling for some of the calculations, and here I'd be delighted to have some feedback from the audience about, the units that we've used to make some comparisons to previous data in the literature. So here we actually see in the blue is the stretch, and in the orange is the power versus time. So here we have and this is for the passive muscles at L naught. So here we are at L naught, and we stretch by 5% L naught, and that's the blue line. And what you can see is the total power. So that's in the K1B1 plus K2B2. So the K1B1 is shown by the dotted line, and the K2B2 is shown by the dashed line. And so what you can see, and so this is for the passive muscle, and then when we activate, you can see this is like joules per second times 10 to the minus 4, and this is actual joules per second. So we get a really big increase in the power when we activate the muscle, in the power of the viscoelastic transmission. And most of, but not all of this power, particularly in the active muscle, is in the K2B2, in the dotted line. And because the K1B1, I'm sorry, is in the K1B1, because the K2B2 gets much stiffer when we activate. So the total power during stretch in the sartorius increases from about like 1 watt per kilogram in the passive muscle to almost 100 watts per kilogram upon activation. And for reference, the sort of the standard force velocity relationship predicts that the maximum power of a muscle is about 250 watts per kilogram. So that's a substantial amount of the total stored in the viscoelastic elements. Now we can compare, over here we have the same data from the sartorius muscle, and here now we have our semitendinosus muscle, which interestingly is much less stiff. So in the sartorius, the power increased from like about 1.5 to almost 100 watts per kilogram. But in the semitendinosus, it increased from, about 8 watts per kilogram in the passive muscle to almost 200 watts per kilogram upon activation. And so I think it's interesting that the increased stiffness of the sartorius doesn't necessarily translate into higher power. And then finally, we can look at the energy storage and dissipation. So, so here again, similar graph. Here we see the total stretch in blue, and the energy in joules in the orange, with the solid line being the total in both viscoelastic elements. So this is the passive and this is the active, and you can see that in both the passive and the active, more energy is stored in the PVK1, but when we activate, because the PVK3 gets so much stiffer, almost all of the energy and strain is stored in that K1B1 or PVK1, PVK2 element. And so it was really a challenge for me to figure out, and I don't know if Natalie's here today, but it was really a challenge for me to figure out units to compare energy storage to the literature. And so I decided to use the Linari et al. 2003 paper as the basis for comparison. So in 2003, Linari published a paper with Roger Woolidge and Nancy Curtin, where they actually looked at single fibers from frog tibialis anterior muscles. And they found that the total energy stored during stretching was up to 0.092 P naught times L naught. So P naught is the maximum isometric force, and L naught is the optimum length. And so the units of P naught times L naught look something like a volume, right, or a muscle mass. And they measured this by actually using thermopiles to measure the heat production in the muscles as well as the work done by the muscles in stretching. And what they wrote at the time was that only about a third of this energy could be explained by cross bridges, thick filaments, thin filaments, and tendons. And so 2/3 of the work was unexplained by any mechanism. And the interesting thing is in the sartorius, and I'll show you some data from the semitendinosus in a minute, the energy stored in passive muscle was only about 0.00034 P naught L naught. But look, when we activate it, increases to about 0.0632 P naught L naught, which is it's kind of scarily close to how much energy storage was missing in Linari's study. And when we compare that, okay, somebody's got a question. Manny's. So Manny, it was the internal tendon in the muscle fiber. So he's talking about, I should say maybe, like the ECM. Maybe he called it a tendon, but I agree that tendon maybe isn't the greatest word there. But I would say, let's just say parallel elastic structures in the single fiber. Good point, though. Okay. So, and in comparison, and again, these are just single individual muscles, and there will be a range when we look at all, 7 to 9 muscles that we've looked at so far. But in the sartorius, it increased from 0.0011 in passive muscles to 0.054. So anyway, it looks like the viscoelastic structures representing titin in muscle sarcomeres are storing a substantial amount of energy. Okay, so muscles store significant elastic potential energy. K1B1 stores more than K2B2 in passive muscles. And that passive muscles dissipate the energy relatively fast. Because the K2B2 stiffness increases hugely in active muscles, strain and energy are concentrated in a K1B1, and the time constants increase tenfold with activation in the K1B1, but decrease in the K2B2. And so, active muscles can not only store significant energy, but they can store it for a significant amount of time. It's certainly for like a whole cycle of gait for most animals. And I don't know if we looked at elephants, they might even have larger time constants. All right. So, it's possible that that the viscoelastic energy stored in these, or I should say, the elastic energy stored in these viscoelastic structures representing titin might account for Linari's missing energy, but that's making a lot of assumptions. Because we so far haven't measured, the total energy from ATP hydrolysis that's contributed in these frog muscles. All right. So I could stop here, but I'll just maybe briefly say that energy storage is severely compromised in the MDM muscles. They can store about 10 times less energy in the active muscles than the wild type. Whereas interestingly enough, due to expression of genes, I should say, due to expression of exons in the K2B2 part of titin in the 112, 158 transgenic mouse, the phenotype is restored. And lastly, we're pretty excited about this type of analysis because we think that for any mutation in cardiac or skeletal muscle, if we know either the PVK gene expression or the viscoelastic properties from system identification, or both, we can estimate how these changes in titin or other sarcomeric proteins, including like myosin binding protein C or actin or myosin, might impact the energy storage in muscle. So the last little part is striated muscles are adaptable and efficient. Every movement cycle is different because the external loads applied to the muscles are different, and the viscoelastic properties depend on strain and activation, which provides embodied mechanical intelligence, and this can reduce the energy and energy cost and computational burden of control. Muscles are efficient. They can use energy from ATP hydrolysis as well as energy from applied loads like stretching. They can store this energy across multiple timescales to amplify or dissipate cross-bridge forces and power. And in evolution, individual titan domains appear to have been selected over millions of years, hundreds of millions of years, to provide different specialized properties over orders of magnitude of frequency and force. So thanks for listening, and I'll stop sharing, and I'd be happy to take any questions.
I'll ask a question. I guess I'm one of the only folks who've known you for more than 20 years, maybe on this call. My question is, why not compare it to an estimate of muscle work in the units of joules per kilogram, which I think is somewhere around, it's a big range, let's say 60 to 150 joules per kilogram, let's say. And that's even a pretty high estimate because it's assuming infinitesimally small, short velocities and a very large amount of shortening.
Yeah, so I could actually do that really, really quick right now if you want.
Yeah, because exactly, because you could do your P naught times L naught and
okay, yeah, because so I did, since I did the watts per kilogram, I have all the data right here. So let's just, so this is for the semi. So that's a joules. And the kilograms is 19. Uh-oh, Manny froze. Maybe I'm frozen.
Your video is
hear
frozen,
you.
but your audio isn't.
I froze, but yeah, 19.65 for the
That's a good number, I think.
yeah, it's not trivial, right? Okay, and then we can do it for the sartorius, so that's going to be let's see. $15. So 15 to 20. So not so big that it's everything, but not so small that it's
Insignificant.
Yeah, negligible. Yeah.
Yeah. Very cool.
And of course, this is a small amplitude stretch at low velocity. So, Linari et al., that paper says that and this is single fibers, I mean, there's a lot of assumptions, but that paper says that they got the most energy stored at about like 1.6 L naught per second. So, yeah, so anyway, I think, I think that this analysis shows that, this is it's a substantial contribution. Yeah. Other questions? I thought, Manny, I'd be interested too in your opinion, but I thought the power amplification was also really reasonable.
I agree. Those numbers seem totally within reason. I was going to ask you one thing. The time axis was the same for both the data sets you showed. It's in seconds, yeah.
Yep.
So it's
yep.
not transient. That's the cool part, is that, yeah, you can get transient expressions of power that are pretty high, but over a very, very short time scale. But I think this is over a time scale that's actually meaningful to an organism's movement.
That's right. That's the point I was trying to make, that muscles, certainly for the mice and the frogs, the muscles can hold on to energy for a whole gait cycle. Yeah. Yeah. Yeah, so and for the in terms of the modeling studies that were talked about yesterday, we hope to, get the model working soon, and uh, then apply it to people's data.
Well,
So go ahead.
I think it's exciting to have the addition of the muscle tendon unit prep, where we know there's an additional spring in series at the end of it, because you could easily tag sort of the two body and another sort of spring in series with them, or some kind of additional configuration that that should show up.
That's right. That's right. So for those of you who don't know, one of the summer research projects is going to be done in Manny's lab. And so he's going to do these ramp stretch experiments in situ rat medial gastroc muscle plus tendon. And so we expect to have a 2 Kelvin Voigt bodies in series with a spring model come out of that. And what we can do is then we can really compare, we'll be able to compare what's the power amplification by the muscle, what's the power amplification by the tendon, how do they match up, quantitatively. Savannah's question in the chat was, would the 2 Kelvin Voigt model be able to fit most types of muscle? And it seems like, for pure muscle with no tendon attached, which is the way we did our experiments ex vivo, yeah, we expect to find that the 2 Kelvin Voigt bodies should apply. I mean, 2 out of 2 frog muscles and 2 out of 3 mouse muscles, and the mouse muscle that it didn't fit kind of is the exception that proves the rule, because that has been genetically engineered to not have one of the elements. Yeah, so I think it's going to be fun to apply this model to more muscles. Also, as I tried to intimate, I think that this is the simplest possible viable model given what we know from previous studies. So it's really interesting that the evolution kind of hit on the simplest possible model. But I think how those elements in Titan have evolved over evolution is going to be another really interesting aspect of research going forward, to look at like why not elephants? You know, we do
So
expect some interesting scaling rules. Go
I have a
ahead.
question kind of on time scale. So right now we're going to look at ramp stretches, right? And the ramp stretches show us some sort of perturbation like, you know, behavior in legged locomotion. But what do we do about bigger time scales, right? Like how does this apply? What's the next step after looking at ramp stretches? Because like that's only a very small time scale in dynamic locomotion. So like where would we go from here?
Yeah, that's a great question. So there's two reasons. Well, so, all right. So where we're going is the full-scale Lagrangian dynamics model. And there's two reasons for that. So one is like the whole system identification approach is to do a linear analysis at an operating point. But what that system identification shows is that muscles aren't linear. Muscles are super nonlinear. They have exponential stiffness, not linear stiffness. They aren't Hookean springs. And so to capture the dynamic behavior, you need a nonlinear model. The other interesting thing, and I think this came up in an earlier, maybe it was an all hands meeting when we were talking about modeling, but each ramp stretch experiment only probes a limited range of stretch velocities, right? But the interest, and we know that stretch velocities can occur, or stretch can occur at very high frequency during, like natural perturbations, as, you and Monica have, measured in, and others have measured in guinea fowl, and, like up to 40 hertz. So, but the problem is that when you do ramp, like, so one question would be, should we do more ramp stretch experiments with different stretch amplitudes and velocities? And the answer is that because of the cross-bridge force velocity relationship, the properties that you're going to get out are going to be, the properties of the whole system and not just the titan. So we think that by just measuring, so we're just probing what's the stiffness at this length and activation. And we think that, those values should be, should allow us to make this dynamic model. And then the dynamic model should be able to accurately predict what happens with different stretch amplitudes and velocities. And we can validate the model by comparing the model predictions to the data that we have. And then if those predictions aren't good, then maybe it's back to the drawing board and we need to add parameters that would account for that. But the intuition is that the model will take that into account. So when you stretch a muscle fast, the cross bridges, if you stretch it fast enough, no cross bridges are attached. And so then you're not going to have a cross bridge just winding the titan. You're going to have different active properties because that winding isn't occurring. And yeah. Did that answer your question?
Yeah, no, it answered my question. I'm just thinking and how could I apply this to my own research where I'm having problems explaining power amplification between in situ and ex vivo research, right? Um,
Yeah.
but we're steps away from it. I'm just thinking into the future, how it would
Yeah.
be applicable. But right now, these stretch amplitudes are all across the same frequency of stretch shortening cycles, correct?
So we didn't do any stretch shortening cycles. So these are just ramp, stretch and hold
Oh,
on
just ramps. Oh, there,
plateau.
yeah,
Yeah.
okay.
Yeah. But we have lots of data sets. So first thing is, we super want everybody in IMSI and lots of people not in IMSI, to have the model. So, so Anne and Jordan are first in line to get the model. And so as soon as it's done, Rick is planning to go visit them in Golden Colorado and get them up and running with it. But, you know, when I was in Boston, you know, I talked with Hugh Herr, and they have some really interesting data sets that we want to apply to the model. And the model, you know, it will be like a hill model. So you will, the user will, you know, input length and activation, and the model will tell you the time varying force. And what we'll have is like a spreadsheet where you can specify, okay, like this is a mouse soleus, or this is a frog semitendinosus, or, you know, we could potentially use gene expression data, or you know, CISO data, or just wild guesses to, you know, potentially do, you know, the human soleus or the human, you know, biceps brachii. I think it's really comforting, and came as a surprise to me that the mouse and the frog data were so similar, given like the 20-fold, you know, differences, you know, the big differences in size, you know, big differences in species, big differences in locomotor style. And so that gives us some hope that we won't have to make you know, parameterize, you know, all of the parameters differently for every single type of muscle. But there's one really interesting thing that I didn't have time to talk about, and that's the difference between fast and slow muscles. And so we have done, in addition to, you know, the different genotypes, we've also done mouse EDL and mouse soleus. And the really interesting thing is that they have the same PVK structure, but the EDL, like the identical exon expression in PVK, but the EDL is stiffer, right? And the EDL develops force faster. And so what's going on? And it turns out that the position of the N2A clutch is different. So the clutch is closer to the Z disc in the EDL than it is in the soleus. And so basically we think that there are two knobs in Titan that evolution has mainly depended upon to change the muscle stiffness. And that's like how far from the Z disc is that N2A, and how stiff is the PVK. And so, and when you think about like the Lagrangian dynamics models of fast versus slow muscles, so you're going to have you're going to have on the one hand, the faster myosin, you know, faster cross-bridge kinetics combined with the stiffer Titan. And what that's, and actually also like the variable transmission ratio that you get from the winding at different locations relative to the Z disc is a thing. So what happens is it looks like, you know, and Rick could chime in here, but from the mechatronics perspective, it looks like the fast muscles are running in high gear, right? And the slow muscles are running in a lower gear. And it turns out that similar differences occur in Titan isoforms in the heart. And so I think it'll be interesting once we have the model, we'll also be able to look at how differences between cardiac and skeletal, how moving these different things changes the energy storage. So I haven't done the energy storage calculations for the fast and slow muscles, but I think offhand I would say like the energy storage that is going to be greater, but the ability to hang on to that energy is going to be less for the fast muscles compared to the slow.
What Kiisa was talking about, this is Rick Kessler. hear about the fast twitch and the slow twitch. The fast twitch, the titan connects to the thick filament at a shallower angle. So when you spin up the thin filament, the pitch of the helix is longer per rotation, which means you're running in high gear. So the fast twitch needs more torque out of the cross bridges because it's running in a high gear, and so that's why it's able to go so much faster and why it wears out faster, because we've modeled the energy consumption, both at the cross bridge and in the Titan, and it'll just eat energy, when you're trying to really execute a high bandwidth command, which is what the fast twitch is there for.
I guess again, did I stop sharing? Yes, I did. Yeah. And, just going back, Manny, and Natalie's not here, but we'll have to have a conversation with her about this too. You guys have data, looking at, how if you change the tendon compliance. Oh, and I think Greg Sawicki has some data like this too. So it'd be really interesting to try and model those data. So yeah, so once we get this model going, I'm not going to have to ever do another experiment. But yeah, we do want to disseminate it widely, and over the next 2 months, few months, I hope they'll become available to people.
Yeah, my student Miles has nailed down this injectable cross-linking agent that pretty much can almost double tendon stiffness.
Yeah, that's cool.
So it's
I
a
have
nice
some data
correlation.
yeah, I have some data from Henk Granzier that I haven't analyzed yet, and his postdoc Songwon Han, but they're also drugs that cross-link PVK and make it stiffer. So I think that that'll be another way to test, to test the hypothesis: do these units really correspond to PVK Titan? So. Okay, well, I know what I need to share my slides again because I know what I did that I forgot to do. So let's just go on to the next one. So here we are back at the QR code. So if you're a trainee and you didn't sign in earlier, you can do that now. Love to have your feedback, since this is kind of the first time I've presented both the sort of the viscoelasticity primer at the beginning, as well as the energy and power results. So I'd love to have your feedback. And otherwise, we're almost out of time. And don't forget that Monica is going to be talking about dimensional and non-dimensional analyses next Thursday at the same time.